Can you see the screen, Aisha?
Yes, sir.
Perfect. Okay, so we started with molecular basis of inheritance. And would you like to just quickly give me a quick recap? Because I think others will join late, or maybe so we should keep going. So how about you just tell me a brief overview? Don't have to go into details if you don't want to. But what did we do in the last class? And any part that you are not clear about, we can go over it again.
Yes, sir.
Yeah.
So we studied about the genetic material. So any cell that has a nucleus contains DNA, and DNA can replicate, and it can convert into RNA by transcription, and RNA can convert into proteins by a process called translation.
Okay.
And this is called the central dogma of biology.
Yeah. And also you would like to remember that why we call it central dogma, and what's happening in central dogma is basically it's a transfer of genetic information, right? From one kind of a molecule to another kind of a molecule, finally reaching to proteins, because proteins are the functional units that are expressed in the cell, and then any function or structure takes place, right?
Yes.
Yeah, great. Continue.
Then we learned the functions of proteins like cytoskeleton, enzymes, and receptors. Then we also discussed the difference between DNA and RNA. We learned about the scientist Frederick Mister, who discovered nuclein inside the nucleus of the cell, which is an acidic molecule. And James Watson and Francis Crick discovered the double helical structure of the DNA.
Right. Yeah, we drew some of these structures. Is it clear to you? Good evening, Alia. Alia just joined in. Am I audible to you, Alia? I'm not sure if you can hear me. Good evening, Iram.
Good evening, sir.
Okay, great. I'm at least audible to someone. Okay, Aisha, yes. So Aisha is giving a quick summary of the last class. Yes, continue, Aisha. We talked about the double helix structure, right? Yeah. Yes,
So
Iram.
can you please check if the spotlight feature is on so that I don't have video?
Yes, Krish, just wait, give me 1 second. 1 second. Yes, it is. Because the option is to remove spotlight, which means it is already on. Okay, great. Yes, Iram, go ahead. Aisha, we are talking about the double helix structure. Yes.
Yes, so we learn about the backbone, which contains of sugar and phosphate. And we also learned about the different nitrogenous bases, like adenine and guanine, which are the purines. Purines are double ring structures, and thymine and cytosine, which is pyrimidine, and there's single ring. We also learned about nucleotide and nucleoside. Nucleotide is a monomer of DNA. And we learned about its structure with double bond and triple bonds.
Right. So the double bonds and the triple bonds, basically the hydrogen bonds, are between the base pairs. Okay. Yes, good to see you, Ram. How are you doing?
Good. So I'm actually back from India.
You're back from India, or you're back in India? You're back from India.
I'm back to Doha. Yes.
Oh, you're back to Doha. Great. That's okay. That kind of explains some good internet connection. Though I'm in India and I have good internet connection most of the time. So yeah, it is. Okay, anyways. But I was just like, I think I believe that when you visited India, you were visiting your hometown and, where were you visiting? Like you were in cities, or you were in.
So 3 places actually. First, I was there in Bombay for probably 2 weeks. Then I went to Uttar Karnataka, and finally I was there in Bangalore.
Right. So I think these places, I don't know about Uttar Karnataka, but Bangalore and Mumbai has good internet connections because I've been to both the places myself. Okay, anyways, so we're back and Rabi has also joined in. Good. So Aisha is talking about the nucleotide and nucleoside that we learned about. And I shall just wrap because this is where we ended in the last class, right? And I gave you the homework to go and read about the salient features of double helix structure of DNA. But if anyone has any doubts regarding this particular thing, this structure here that we made, so remember that DNA has two things. It has a backbone. The backbone goes in a spiral fashion, just like a spiral staircase that you will see in some vintage home or some places you have these, spiral staircases. Exactly like that, it's a DNA. And the backbone, which is the handrest which you use when you're climbing the stairs, that is all made up of sugar and phosphate. And the steps, basically, you can understand are the bases. Now, I told you that there are two different kinds of bonds that takes place between bases, double and triple. And that depends on which base is pairing with which. It is very, very specific. Adenine always naturally pairs with thymine. Guanine naturally pairs with cytosine. Adenine does that with thymine through a double bond, and guanine does that with cytosine with a triple bond. Now, these are just facts that we can read in books. The logic behind that is the structure of adenine, guanine, cytosine, and thymine. Since it is not very, very important for you to remember these structures, but as an alternate take home assignment, I'm giving you to go back and look at the structure of nitrogenous bases. Okay. You already know that purines have a double ring structure and pyrimidines have a single ring structure, but just go back and look at the structure of these nitrogenous bases. And you will also on internet very easily find what is the structure when adenine makes a bond with thymine and when guanine makes a bond with cytosine. You will understand better that why adenine cannot form a bond with cytosine or guanine cannot form a bond with adenine or thymine. Okay, why it is so specific. So it's just good for you to know so that this stays in your mind for longer. Now, how many of you went back and read the salient features of the double helix structure that was suggested by Watson and Crick? Anyone did that? Basically, we talked about most of these features, and this is one question that is kind of a very common question that comes in 2 markers, 3 markers, even 4 markers. Can even come in 5 markers because there are basically 5 to 6 salient features. And it's very easy. So if we start with this, you have understood by now that there are 2 polynucleotide chains, right, in a DNA. When I make a DNA, let's say we do this, right? So this is one chain and I call it polynucleotide because I just told you in the last class that nucleotide, this structure, nucleotide contains phosphate, sugar, and bases, all the three. So nucleotide is a monomer. DNA is nothing but a very long molecule of these monomers attached to each other, like a chain, okay? Like the chains that we see in real life, exactly like that. So the first salient feature that Watson and Crick told the world about is that DNA is made up of two polynucleotide chains, okay? So there are two polynucleotide chains where the backbone is sugar and phosphate, okay? Can write down the backbone is sugar and phosphate, sugar phosphate. And base as a molecule. Okay, am I audible and visible, everyone? Am I audible and visible, people?
Yes, sir. But your voice is kind of breaking.
Okay, I think okay. Is my voice stable, people?
Yes,
Yes,
sir.
sir.
Okay, great. I think it should be stable now. So let's go with my audio for the time being. Sorry for this. Just talking about internets, and I think it listens. Okay. Let me know if you can see the screen.
Yes,
Yeah.
sir.
Can you all see this? Okay, perfect. And also let me know if my voice breaks. Okay, I'll try changing the network. It's okay for now, right? Shall we continue?
Yes, sir.
Yes, sir.
Okay. So the first thing that's not very difficult to remember is obvious thing that you know, that there are two polynucleotide chains. The backbone is sugar phosphate, and the bases basically they project towards inside. Okay, like the stairs. The second thing is these two chains that we are talking about, this is the chain 1 and this is chain 2, they actually have very opposite polarities, or we can call it anti-parallel polarities. Now, what does it mean by anti-parallel is this. So wherever you have your phosphate, remember, we call that site as 5 prime. Okay. And wherever you have your this sugar, and let me here remove this phosphate to make it clear to you. So when we have a sugar, at the end where this phosphate bond forms, there is a OH. So basically here was a OH, here was a OH. So this OH binds to the phosphorus molecule. So wherever this OH moiety is there, we call it the 3 prime end. So we also call it 5 prime phosphate end. Towards the 5 prime is phosphate, then comes sugar, and 3 prime hydroxyl. Don't worry if you don't remember this, it's completely fine. Just remember that phosphate is towards the 5 prime and sugar is
So your voice is still cracking.
okay. How's my voice now? Can you all hear me?
Yes, sir.
Okay. Okay. Fine then. You all can also see the screen that I'm sharing, right?
Yes, sir.
Okay. I apologize for this. I don't know. Both the networks are showing full, but still somehow behaving weird. Okay. So was the above clear about polarity? I think not. So let me tell you about this once again. So the chain that we are talking about, okay, the two DNA chain, the polynucleotide chains, the backbone, they have a Earth, we say from South Pole to North Pole or North Pole to South Pole. Similarly, there is one phosphate end and one hydroxyl end. Okay, does that make sense? Am I audible, people? Okay, do let me know if I'm audible.
Yes, sir, you're audible.
Perfect. Is this better network?
So your voice is still cracking.
I don't know. I think it's some issue because I can't understand what's happening. It's showing a very good speed actually. I don't know. It's very weird. It's showing around 45, 50 Mbps speed. Still, my voice is cracking. Is it better now?
Yes, sir, it's fine now.
Okay, thank God. Let me share the screen again. Let's hope it. Okay, how's it? Can you all see the
Fine, sir.
screen again? Perfect. I don't know. I think it's rains or something. The weird thing is that the connection is showing proper. Anyways, okay. So is this polarity thing? Could you hear what I talked about the polarity of the DNA molecule? So remember, there are two chains in the DNA, and when I was making this structure, we started with phosphate, then a sugar, phosphate, sugar, phosphate, sugar. From every sugar, one base projects inside. So towards the phosphate group, we call it as the 5' phosphate end or the 5' end. And towards the sugar group, there is a hydroxyl molecule, OH, present at the end that will now form a bond with the next phosphate group. So towards the sugar, we call the 3' end or the 3' hydroxyl end. Is that clear? Phosphate is towards the sugar is towards the 3'. The polarity is clear. Give me a thumbs up. Okay, perfect. I got 2 thumbs up, which means am I audible? So both chains have an anti-parallel polarity. So if one chain is prime to 5', so it's like Mariam is saying that is my voice breaking for others as well?
Yes, sir, it's breaking.
Uh, bad news. How about now?
It's fine, so
Yeah, it's sorry, it's frustrating, but I don't know what else I can do. It happens sometimes. I think I shouldn't have talked about internet in the beginning. Anyways, so if one chain is starting from phosphate and going towards sugar, which is 5' to 3', the other will be in opposite polarity. And that makes sense because that's how they can join together through base pairs. So the other one will have phosphate ends towards the opposite side and sugar towards this. So if one has 5' to 3' polarity, the other will have the opposite polarity. Is that clear, everyone? So that's second
Yeah.
feature of DNA. Okay, great. Now, the third feature is that you know that the bases in the two strands are paired through hydrogen bond. Okay. So next question that anyone can ask is how are the bases paired? So bases are paired through hydrogen bond. We call it H bonding or H bond. And you already know that adenine and thymine make a double bond together. 2 hydrogen bonds, and guanine and cytosine make 3 hydrogen bonds together. Okay, and this way a purine always comes opposite to a pyrimidine to form bonds. But remember, one thing is a little misleading here. I told you that there are 2 purines, adenine and guanine, correct? And there are 2 pyrimidines, thymine and cytosine, correct? Yes,
Yes.
no? So can it happen that one purine, which is adenine, we know that it forms a bond with thymine, but can it form a bond with cytosine? Because here also the law is still followed. One purine, which is adenine, is coming against the another pyrimidine, which is cytosine. So can it happen? Do you think it can happen? What do you think? Come on. NCRT says, most of the book says that it always have to be one purine which can bind to another pyrimidine. It can't be 2 purines binding together or 2 pyrimidines. So that we understand. Adenine and guanine cannot bind together. Thymine and cytosine cannot bind together. But what about adenine and cytosine, or guanine and thymine? What about that? What do you think? Come on, you just have to make an informed, logical guess. What do you think? So when no one replies, I think I'm not audible. Am I audible?
Yeah, you are audible, sir.
Okay, great. Sigh of relief. Please don't do this to me, because today internet is also not good. So if no one speaks, I'm like, okay, I'm gone again. See, it cannot happen because I just told you adenine is capable of forming 2 hydrogen bonds. Okay. Whereas cytosine, though it is a pyrimidine, but it can form 3 hydrogen bonds. That's why cytosine always pairs with guanine and adenine pairs with thymine. That's why I'm giving you this task to go and look at the structure. It will tell you a lot of things about this specific base pairing. So apart from remembering that a purine binds with pyrimidine, also remember that that purine will always be adenine. And if it is adenine, then the pyrimidine will always be thymine. It cannot be cytosine. Okay. And why is this important? So write down a question that has come in exam. Why a purine always comes opposite to a pyrimidine? Why a purine always comes in opposite to a pyrimidine in a DNA double helix structure? And write down the answer. A purine always pairs with a pyrimidine to keep the distance between the 2 strands, to keep the distance between the 2 strands of the DNA to keep the distance between the 2 strands of DNA uniform. Okay, uniform. So that a double helix, can be uniformly formed. Is that clear, everyone?
Yes, sir.
Perfect. Okay, now the next thing that Watson and Crick discovered and defined is that now this thing, when they figured out, they worked with the DNA and it was coiled in a right-handed fashion. So the DNA that normally is present in humans, all the animals and plants is called the bDNA. So they also, at that time, they did not know that there are different kinds of DNA coiling in nature. So now we know that there can be three kinds of naturally occurring DNA coilings. One is called the bDNA, then the zDNA, and the aDNA. This is the most common, and the B stands for biological. So biologically, this is the most common DNA, bDNA. Humans also have a bDNA inside. Now by B, I mean that this DNA is coiled. The two chains of the DNA are coiled in a right-handed fashion. Okay. I hope it's clear to you. Like if you just take two straight, take a ladder, okay? Think of a ladder, and then this is your left-hand side, this is your right-hand side. So start twisting it in a right-handed fashion. Like you open the lid of any jar. When you open the lid of a jar from top, you move it from the left towards the right. Sorry, when you unopen, when you close it, when you close the jar, the normal standard scenario is that you are rotating it in the right-handed fashion. Do you understand what I'm talking about?
Yes, sir.
So if it's right coiled, it is for sure bDNA, and the pitch, write down the pitch of the DNA is 3.4 nanometer. Pitch is this one turn. When you turn it, when you coil it, when it takes one turn, it is called a pitch. So the pitch is 3.4 nanometer. And there are 10 base pairs. 10 base pairs in each pitch. Okay, and this tells you that the distance between two consecutive base pairs is 0.34 nanometer. You just have to divide. And the fifth thing that is a characteristic feature of the DNA double helix is when you look from top, so it will look like a circle to you with bases stacked over each other. Do you understand why? When you look from the side, it will look like a spiral with bases going in. But when you look from the top, it will look like all the bases are stacked one over the other. Because this is exactly how the DNA stays stable. So write down the plane of one base pair. Plane of one base pair stacks over the other. Stacks over the other in the double helix. Plane of one base pair stacks over the other in the double helix. And you would like to write, make a star mark and write, this gives stability to the helical structure. this provides stability to the helical structure. And this is also one reason that why DNA is more stable than RNA, because in DNA, both the sides have a backbone and bases come inside. So when they stack very, very properly. RNA, there is some issues with stacking because there is only one backbone. It's a single stranded molecule. So that's also is what makes DNA more stable chemically and does not allow it to react with a lot of molecules that RNA can react with because it's an open strand. Does that make sense, everyone?
Yes, sir.
Okay. Perfect. Now this is about DNA. But let me ask you one thing. How many DNA molecules do you think are present in human body? Or let's make it simple. Let me not ask about human body. Let me ask about one single cell. Let's start with the beginning. Let's talk about a zygote. When a zygote is formed after fertilization, how many DNA molecules are present inside the zygote? Can we count it, or is it uncountable, or what should be your logical guess? What do you think? How many DNA molecules are present in a zygote? This actually is a question. That's why we are discussing it. So think about it. If I would have asked the same question in a different manner, you would have found the question easier. If I ask you, how many chromosomes are present in a zygote, a human zygote, what will you say? Okay, I got an answer in the chat. Mariam says 46. Perfect. So you already know that there are 46 chromosomes in a human zygote, because a human zygote is a diploid cell. Half, which is 23, comes from sperm, and the other half, which is another 23, comes from the ovum. Perfect, 46. And so how many DNA molecules do you think are present in a human zygote?
92.
46. Okay, exactly 46. So a chromosome that we talked about, I kind of got why Iram said 92 is because
So
you
there
are,
are two molecules of DNA in a
sorry?
chromosome. There are two molecules of DNA in a chromosome.
Yes. So what Iram is talking about is this thing. So when we make a chromosome, we always make a chromosome like this, right? The centromere. And I already have told you about this is one chromatid and this is another chromatid, right?
Yes, sir.
Yes. Yeah. So that's why Iram thought that, okay, there are 92 DNA molecules, and she's not wrong. But if I am defining the stage, at what stage I'm asking, that makes sense. That makes more sense to give the answer. So if in any cell, when the cell is not dividing, there is just one chromosome, one chromatid for each chromosome like this, because it doesn't make any sense for a cell to have two copies of every chromosome for no reason. And let me tell you one more thing. Actually, in the nucleus, if this is a nucleus, let's say this is the nucleus. In the nucleus, chromosomes are not compacted all the time. In a normal cell in the body, chromosomes are open. They stays in a region, like this is one chromosome. This will be another chromosome. And literally it happens like this, and this will be another chromosome. So they open up and stays still like they are still coiled, they are still super coiled, but they stay in their own regions. Like this is one region, this is one region. And why are they open? Because in a normal cell, you would actively want transcription and translation to happen, right? You would always like some proteins to be formed. So the transcription machinery should always be able to reach that part of the DNA or the gene. Let's say your gene is here, this part of the DNA. But if it is super coiled and this part is closed somewhere buried inside, that gene won't be able to transcribe. And this actually happens in some cells. You remember at the very beginning when we were starting reproductive system, I asked you a question. So everything in our body comes from a zygote. Let's discuss this for 2 minutes so that this thing becomes very easy for you. Every cell in our body, in my body, every single cell, from the eye to neurons to bone to muscle to my hair follicle cells, everything has come from zygote. Like at one point of time, I must have been a single cell zygote, right? Then I became a multicellular organism. Does that make sense, people?
Yeah.
So every kind of a cell is made from the same blueprint. It contains the same chromosomes, the same genes. Yes or no?
Yes, sir.
Then how does a kidney cell is very different from a liver cell and is a very different from a heart cell and is very different from a bone cell and is very different from a red blood cell? Where does this difference come from? Because the nucleus in the heart cell also contains the same 46 chromosomes, having the same 28,000 genes, and the cell in my liver also contains the same chromosomes and the same 28,000 genes, and same in my neurons. But then why is a neuron and a liver cell a liver cell? Have you ever thought of it? There must be some difference, right? Aisha? Am I audible, people?
Yes, sir.
Yeah, so would you like to take a guess? Why is our liver cell different from a neuron? But it contains the same number of chromosomes and the same genes. Okay, let me ask you a simple question. I gave Aisha and Iram, both of them, the same set of acrylic colors and a canvas and the same set of brush. Okay. But they both ended up making two very different paintings that look very different. Maybe Iram has made a forest, but Aisha has made a sunset, something like that. How did that happen? Your colors were same, your brushes were same, your canvas was same, but still, how did you both manage to make two different paintings? Am I audible, people?
Yeah.
Yes.
So, yeah. Yes, Aisha. What do you think? Why did you make two different paintings? It's a simple question. I cannot simplify a lesson better than this. Please, you have to, you have to participate in this. Otherwise, we can just move on and I can keep telling you facts about DNA. Yes, Iram. Mariam Rabia can also speak. Someone has written in the chat. Okay, that's also good. Rabia says, due to different imagination, creative thinking skills. That perfectly correct. But let's for one moment keep imagination and creative thinking skills separate. Only let's talk about in terms of the color, the brushes, and the canvas. Why did the two paintings came out to be different? I'm sure that what you're saying is perfectly correct. Iram and Aisha are very differently creative and imaginative in their own aspects. But what also happened because of that imagination and that creativity is that at the same time, Aisha chose a different brush to pick up and Iram chose a different brush to pick up. Let's say I gave both of them 1 hour to make something. In the first 15 minutes, Aisha chose a very different set of brush and very different colors. She dipped that brush in and she decided to touch different parts of the canvas to make a sunset. Iram did very different things. She chose very different brushes from that set and she chose different colors and she picked different regions to color. Do you understand what I'm saying here?
Yes.
So it is basically not what genes you carry. Okay. Yes, Rabia says they would have blended the colors. That's perfect. So what matters is not that our liver and heart cells carry the same genes. What matters is which genes are expressed when. So there are certain genes in our body that only express in the heart cells, but they never express in the liver cells. And those genes keep the identity of the heart tissues as heart tissues and differentiate the heart tissues from any other kind of tissues. Does that make sense to all of you?
Yes, sir.
Yeah. So this is called differential expression of genes. All 28,000 genes which are present in our body are not active everywhere. Now, this was just the premise. Why I talked about chromosomes is that how does a cell decides that on the DNA, this is a DNA, let's say this part is gene A and this part is gene B. Now in a heart cell, gene A should be regulated and gene B should not be expressed. So let's say this piece of DNA is present in our heart tissue. So this gene is important, so it should express, and this gene is not required in heart, so it should be silenced. So how can you do that? And I have told you multiple times, the way you will solve a problem is exactly the same how biology solves problem. We are nothing but biology happening inside our bodies. So if I ask you that you have to find a solution so that only gene A gets expressed and gene B stays silent, how are you going to do that? Quickly. Even if you want to give wrong answers, give it, but give it so that we move forward. This is what you are missing in your education, people. Logical thinking, a rational discussion, why it is the way it is. It's also not in your textbooks. In textbooks, it is what it is, where it is, how it is. But why it is the way it is more important to understand. So how will you solve it? Okay, Iram, I gave you a task. Gene A, express, gene B, silence. How are you going to do this? Let me simplify it for you. You made 2 paintings, okay? Now you want some guests appeared to your house and your parents want you to show them your paintings, but you decide I'm only going to show them painting A and not painting B. So what will you do? Sorry? Say again. Right. It's a simple thing.
Just hide the painting somewhere.
Perfect. See, you know all the solutions in life. Why are you not speaking up? You will just hide the painting you don't want to show so that the guests or anyone does not have access to that painting. So that painting will not be shown to them, right? It's a simple logical solution in a day-to-day life. Biology does the exact same thing. When it in a heart cell, when it wants gene A to be expressed, the gene A stays uncoiled. The DNA, this part of the DNA, stays uncoiled and open so that guests, which here are transcription factors or the molecules, can come and copy that gene A into a RNA. But if you don't want gene B to be expressed, coil it, coil the DNA and hide it somewhere in. Now the molecule cannot reach it, right? If the molecules which are important to transcribe a RNA from a DNA cannot reach that part of DNA, they also cannot transcribe it. So no RNA will be formed and no protein will be formed. That way, the gene will be hidden and the gene will be silenced. But the gene is present on the chromosome. It is always present in every cell. Make sense? Aisha, Iram, Mariam, and Rabia, give me a thumbs up if that makes sense to you. Yes. Do you think that biology is very simple and logical, just like you? It doesn't do fancy things. It does do fancy things, but the fancy things, the logic behind the fancy things is very simple. So a gene which is open and that part of chromosome or chromatin which is open is called euchromatin. Okay, and the gene is called euchromatized. Euchromatin, the word eu means true. True means this is a true chromatin that can be transcribed into RNA. And the gene which is silenced is called heterochromatin. This term will come later, but I'm just introducing this term here so that when later it comes up, you understand it quickly. So a heterochromatized part of the DNA is silenced and the euchromatized part of the DNA is active. So the genes present in this part of DNA, which is open, will be transcribed and will be expressed, but the genes present in this part of the DNA, which is closed, hidden, heterochromatized, will not be active. Okay. Now let's come back to chromosome. So normally when the cell is not dividing, there is only one copy of every gene on one chromosome. Just before division, replication happens and the second sister chromatid comes up. So if I ask you how many DNA molecules will be present during metaphase or telophase, then you will say 92 because they're just before division. But if I'm talking about a normal non-dividing cell, at interphase or S phase, you will say 46. In a bacteria, which is the simplest organism, there is only one chromosome. And that chromosome uncoils to form a single DNA thread. So there is only, yes, Iram.
So in S phase, it's like after duplication, right? So there are two DNA molecules, no?
In S phase, there are two DNA molecules. I'm talking about interphase. So before and after, like between two division events, when the cell is resting, it just have, and you only see chromosomes like this in a dividing cell, okay, which has two sister chromatids. Otherwise, you don't see this kind of a thing. So you should know these things. It's very important. Okay. So there are 46 DNA molecules in a normal human cell, your skin cell, your neurons. And do you know how long is one single molecule? Any idea? Let's do some mathematics, okay, and figure it out. Okay, let's start with the very basic thing that we just studied. Okay, things will be simple. First thing first, like in a maths, we take whatever is given. So what we have is how many total, what is the distance between two base pairs in on a DNA?
0.34.
Perfect. Distance between two consecutive base pair is equal to 0.34 what?
Nanometer.
Nanometer. So in terms of 10 raised to the power, what can I write? Nanometer means
Minus
nano.
9.
Yes, 10 raised to power minus 9 meter, because now I wrote this, so I can convert it. I have converted into meter. So 0.34 into 10 raised to power minus 9 meter is the distance between two consecutive base pairs. Now, do you know that how many total how many total bases are there in a human genome? Let's talk about a haploid cell. How many base pairs are there in a haploid human cell? Only one copy. Let's say a sperm or a ovum. Total base
So
pair
which cell?
in a haploid cell, sperm or ovum, anything. Haploid cell means one copy of the genome, right? Just to make things simple, I'm taking one copy of the genome.
Is it 3.3
3.3 what?
into base pair? Is it 3.3 into 10 to the power minus 9?
Minus 9 is, you are counting now. Minus 9 is like, in length, you can say minus 9, but how can you say that I have 2 raised to power 2 into 10 raised to power minus 2 mangoes? You can't have that, right?
Okay.
So this you are giving me in terms of numbers, total number of base pairs. So numbers cannot be in minus. Real numbers are never in minus, right? Length is in minus because we are converting a nanometer into meter. Okay. Either you convert in terms of number also, but numbers is just discrete. I got an answer in chat. Let's see what is that. Rabia says 10 raised to power 9. What is 10 raised to power 9 called? It's called a billion. So we have 3.3 billion base pairs in a haploid cell. Total all the 23 DNA molecules combined together, because there are 23 chromosomes in a haploid cell. So if I take all the 23 chromosomes and I count all their base pairs, there will be 3.3 billion base pairs. And how do we know it? Actually, we have counted. We means scientists have counted each and every base, because there was a big project that happened about which we will study, which is called Human Genome Project. It took around 13 years on paper. Actually, in reality, it took more than that. It took 13 years on paper to know the sequence of every base pair in a human genome. Okay, so we know that there are approximately 3.3 billion base pairs in a haploid cell. And the distance between two base pairs is this much. Now, if I convert this also for sake of clarity in mathematics, can I write what Aisha also suggested? 3.3 into 10 raised to power 9 base pairs. Now, all I want, how do you find distance in mathematics? If I want to find total length of a haploid genome, which means n. So what will I do? I will multiply 3.3 into 10 raised to power 9 by 0.34 into 10 raised to power minus 9. And you know enough maths to understand that this will get cancelled out, right?
Yes.
and you will be left with 3.3 into 0.34. What is the answer? 3.3 into 0.34 is
1.1.
approximately 1.1 watt. The unit here was meter, so 1.1 meter. Wow. In one sperm, which is so tiny, you cannot see it with your naked eye. Can you imagine that in one single haploid cell, there is around 1 meter long DNA sitting inside the nucleus? Do you think it's possible even? Mathematically, it makes sense because we just know the distance between two base pairs. We also know the total number of base pairs. So the distance comes out to be 1.1 meter in a haploid cell. So what will be the total length in a diploid cell? Total length of DNA. Oh, I have not written DNA here, sorry. So what will be the total length of DNA in a diploid cell, in any normal cell of your body? Around 2.2 meters?
Yes, sir.
Can you see your skin cell with your naked eye? One single cell of your skin. What is the average human height? Around 1.4 to 1.6 meters is I think is your and mine, like all our heights fall into this category if we are average humans. So a human's height from tip to toe is around 1.4 to 1.6 meters. And the DNA, which is the length of the DNA, which is present in each and every cell of that human body, is 2.2 meter. And now if we do a mathematics, what is the total length of DNA present in one human's body? We have trillions of cells. Now, if you multiply 2.2 by trillion, you will be surprised to know that the total DNA length present in one human body will surpass the length of a solar system. It's bizarre, right? But it's true. So single human has, if I keep all the DNA present in one human cell, which practically is not possible to do, but if I keep it end by end, it will surpass the length of our solar system from Sun to Neptune, and not just one time. This is what is even more bizarre. I do not exactly remember the number, but multiple times it can do that journey. Sun to Neptune and back to Sun and Neptune. Now, what do you think? This is mind-boggling, right? Have you ever thought about it while studying chapter 6 in your schools or in textbooks?
Yes, sir.
Now, the next question is even more important. If this is true, which mathematically it turns out to be true, and if by depending on mathematics, we have created bridge and buildings and spacecrafts and everything, mathematics is correct. How do you think a single tiny cell holds 2.2 meters of DNA? One of the biggest cell in a human body, apart from ovum, is RBC, right? And RBC is around how much? How big is an RBC? Around 7 micrometer, approximately 7 to 8 micrometer. That's also micrometer. All the cells will be smaller than RBCs. Most of these cells will be smaller than RBCs in our body. How do they contain 2.2 meters of DNA inside their nucleus? So this part is what puzzled biologists for a very long time. They thought that either what they are doing mathematically is incorrect, or they have not understood DNA as a molecule. But then people found out, no, you are right. People did this in bacteria. Okay. So they took E. coli bacteria and they sequenced the DNA. They found that it has this many base pairs. And then they multiplied those base pairs with the distance between two consecutive base pairs. And they found that the length of a E. coli DNA is 1.36 millimeter. 1.36 millimeter is still very huge because you cannot see a E. coli bacteria with your naked eyes. And it has just one chromosome. Now they started asking, how does E. coli fit this long DNA inside its own cell? And if we understand that, we will also understand how humans and every other organism on the planet does that. Okay. So the answer was DNA is super coiled and highly packed. Okay. Just like you know of something similar at your homes. Can anyone give me an example? I'm trying to draw it.
Ted.
Sorry, what?
Ted.
Come again. I could not understand the word.
Ted, sir.
Can you write it or spell it? Thread, you are saying.
Ted. Yeah.
Okay, great. Yeah, a spool of thread. So in your own house, you will find a spool of thread with your mother's, which is like on a very tiny thing. You have a very, very long thread. If you open that spool might cover your whole locality. It could be like half a kilometer or 1 kilometer long thread, but it's multiple times just packed one on the top of other, right? Something like this. It looks something like this. Sorry for my bad drawing, but you just, a spool of wool or a spool of thread is something you have at home. And that's exactly how biology does it. So it highly supercoils and packs DNA to keep it in a very small space. Okay, that's called packaging of the DNA helix. Okay, so let's go and talk about packaging of DNA helix or DNA molecule. Now, just like a spool of thread where you need a base on which your thread will be spooled, right? Thread is coiled. So in DNA, that base is provided by proteins, and those proteins are called DNA binding histone proteins. Okay, so write down. First of all, before you write down, let me also bring this point forward. Remember what was the charge of DNA? DNA is a positively charged molecule or a negatively charged molecule. What is it? Let's go up. See here, DNA is a, we did not write that. Okay,
Sir.
cool. Okay, my bad. So DNA is a negatively charged molecule. Remember that because of the phosphate group that's present. Phosphate is PO4 minus. Okay, so DNA is negatively charged molecule. That's why it is. Oh, I think we discussed this when we discussed acid, didn't we?
Yes, sorry.
We did, right? Didn't I tell you that how does the acid behaves? So let's take an example of CH3COOH, and when it will become CH3COO minus, it is acetate and it releases H plus. That's why it's called an acid, right? Because it releases H plus. So it itself become negatively charged. So DNA also is a negatively charged molecule because it's an acidic molecule. It releases hydrogen, and this phosphate moiety then makes DNA negatively charged. Is it clear?
Yes.
So if DNA is negatively charged, because there is no, no person or no being that can hold DNA and spool it over a base. So it has to do it by its own self. and chemistry works on some rules. So if DNA is negatively charged, what I need the proteins to be so that DNA can bind on
Positive.
its own. Yes, we need some positively charged proteins. So DNA is bound on positively charged, write down, positively charged proteins positively charged proteins to make it a compact packing. To make it a compact packing. Okay. Now there are two kinds of organisms on our planet. One is prokaryotes, the other is eukaryotes. I took an example of E. coli. So bacteria are prokaryotic organism. And what is characteristic feature of a prokaryotic organism? Why do we call them prokaryotic? Basics, brushing up one. Tell me. Why is a
They
bacteria
have
called
whole
prokaryotic?
nucleus.
They have what? Sorry. They do not have a well-defined nucleus, right? They actually do not have any boundary conditions where you can say that, oh, this is a nucleus. For that matter, prokaryotes do not have any membrane-bound organelles in their cell. That's why they only have one organelle in their cell, which is called ribosomes, because ribosomes are non-membranous cell organelles. Okay. So in a bacterial cell, DNA is just floating around, but it's not scattered completely. It is still compacted. And how it is compacted in a prokaryote? By similar positively charged proteins that hold the DNA in a dense region called nucleoid. Okay. So if I draw a bacteria, let's say this is a E. coli bacteria. And inside, this is the DNA thread, but the DNA will stay somehow locate like in this region, because in this region, there will be many proteins. This green region, what is marked in green is positive proteins that bind with negative DNA and keep it in a dense region. So this region becomes denser than the rest of the cytoplasm. And since there is no, even though there is no membrane, we call that region as not nucleus, but nucleoid. Is it clear, everyone?
Yes, sir.
Now, in a human, because this is what happens in prokaryote, but in eukaryotes, we have a well-defined nucleus. So write down, in eukaryotes, the organization of DNA is much more complex. In eukaryotes, the organization of DNA is much more complex. And is done by positively charged basic proteins called histone. Basic means they are bases, they are not acidic. So that basic. Positively charged basic proteins called histones. Histones. So histones are positive, they are basic, and they bind to DNA. So they are DNA binding proteins. And they act like the base of the spool. Okay. So I'll just make a simplified diagram. let's say this is a histone. Okay. Now histone is made up of 8 single units. So I'm making 4 here. And on the opposite side also there is the other 4, right? You understood? If I number it, 1, 2, 3, 4, 5, you can't see it on the other side, 7, 6, 7, and 8. Do you understand the structure in a 3D?
Yes, sir.
Yes,
So these 8 molecules together are called histone octamer. Very simple. Octa means 8. So in a histone octamer, there are 8 histone proteins. Together they make a ball-like roundish structure on which what I'm drawing in blue is a DNA. So a DNA comes and comes around and goes and comes like this. Okay. You understand what's happening? So this is your DNA helix. Do you understand what's happening here, people? Your DNA molecule is wrapped around the histone. When it goes out, here also there is I think something wrong happened. Yeah, touched. Yeah, so this is your DNA that is coiled around and then it goes away. And at this point also there is another molecule, again another histone. So this is histone octamer, and this is called, yeah, this is called H1 histone. This is like a clipper. Okay, it clips both the ends of the DNA. And this is your DNA molecule. Is it clear?
Yes.
So DNA molecule wraps around the histone octamer, which is also in some books written as core histone proteins. So you can write it down as core histone proteins. They are actually 8. And then 1 is at the clipper. And it continues to happen again. So this goes further. Let me draw this. And then again, after some point, you will find another octamer. Okay. And your DNA will go and again bind 1, twice, and will come out. And again, you will have a clipper molecule, which will clip these 2 ends together. And same thing will continue. Do you understand? Everyone, do you understand?
Yes.
Okay. Now this one structure, which includes H1 histone, histone octamer, and the DNA wrapped to it, is called a nucleosome. Nucleosome assembly, or simply nucleosome. Okay. And with very high precision research, we have figured out that, and it was asked in NEET, so remember, that in a nucleosome, the DNA goes around the histone octamers around 1.6 or like less than twice, or is it 2.6? Let me. So it's exactly 200 base pair that goes. So yeah, right. So from this point, from this clipper onwards to this clipper onwards, the total DNA which is bound on a nucleosome is 200 base pairs. So one nucleosome can wrap 200 base pairs of DNA in it. So similarly, if there are, let's say, 20 nucleosomes, so you have to do 200 into 20. Plus, you also have to add this region, this free region. This is called the free DNA. Okay, and any gene that falls in this region is active. It can't get silenced in this kind of a packing. So this is not the only way to pack. This is the first stage of packing a DNA. We are making DNA very, very short here because we are wrapping it around 2 times in every nucleosome. So DNA will become small, but still it is way big. So we have to go to another level of packing. So after we make nucleosomes, these nucleosomes together form chromatin. So these repeating units of nucleosomes together form a chromatin. And chromatin is known as a bead on string structure because when we look under the microscope, it looks something like this. See, so let's say in an electron microscope, a very powerful microscope, I'm looking at chromatin. So I will see DNA and then a bead-like structure, DNA and a bead-like structure, and a bead-like structure. Can you tell me what is the bead-like structure here? So these beads are nothing but nucleosomes, right? Multiple nucleosomes. Do you understand?
Yes, sir.
And this whole molecule now we call as chromatin. And from here the words come euchromatin or heterochromatin. Because this chromatin again packs. So this chromatin packs like this on top of each other and become very dense. And this is what you see in a chromosome. So when you see a chromosome like this, what you are seeing is a densely packed chromatin. So this chromatin will open and then will open the nucleosome and then you will see the DNA. Is it making sense, everyone? Yes.
Yes.
So chromatin, write down chromatin is also known as bead on string. Structure. Okay. So histone assembly, histone octamer, H1 histone, chromatin, nucleosome, everything is clear to you?
Yes, sir.
Yeah. Now, histones are only responsible to make nucleosomes. Once we make nucleosome and we make a chromatin, the further assembly, the further packing of a chromatin takes place again by another set of proteins, but this time they are called non-histone chromosomal proteins. So for chromatin, write down, chromatin is further packed and compacted by non-histone chromosomal protein, because now we have reached the level of a chromosome. We are making a chromosome. We call them non-histone chromosomal proteins, or in short, some books write it as NHC proteins. So if they ask you in an entrance, which protein is responsible for the fundamental packing at the first level? So it is histone. At the second level, it is NHC, non-histone chromosomal proteins. Is it clear, everyone?
Yes, sir.
And these proteins, the non-histone chromosomal proteins, are the ones which form either euchromatin or heterochromatin. Okay. So if these proteins pack a region of chromatin very dense, so if it is densely packed, it will be called heterochromatin. Because you have done so much of packing, it's exactly like when you go on a vacation and you have to pack a big suitcase. You would like to keep the things that you don't want to use during the journey. You only want to access them once you reach your destination at the bottom, right? You keep your things like charger, your toothbrush in some outer pockets where you can just open it and access it. Do you understand what I'm trying to say?
Yes,
Yeah.
sir.
Yes. So if it is loosely packed, which is like the outer pockets of our packing, it's called euchromatin. And heterochromatin, it is transcriptionally active. Euchromatin is transcriptionally inactive. So let's stop here for today, and we'll continue with heterochromatin and euchromatin in the next class. Okay. And also in the next class, we are going to, so you know a lot about DNA now, but remember at one point of time, scientists did not know anything about DNA. And they also did not know that if DNA is the genetic material or something else, how to figure that out. So we'll go back in time and we'll do this exercise of finding out how scientists figured out that DNA is the genetic material and not the protein, not the lipids or anything else. Okay. So far so good. Are you all on the same page with me? See, this chapter is a little bulkier in terms of facts, but I'm trying to go slow so that it seeps into your heads better. Please let me know if you want me to repeat anything else before we go to the experiments where we will be searching what is genetic material. Everyone is clear with today's class?
Yes, sir.
Yes, sir.
Okay, great. So please go back and read from NCERT, revise it before you sleep tonight. Otherwise, whatever went into your head, a lot of it will just come out. So you have to revise and put it in your neurons permanently. And the second thing is, I forgot what the second thing was. Sorry. Yes. Second thing is I apologize for the internet issues in today's class. Hopefully it will not happen in the next class. Take care. Bye-bye.
Thank you, sir. Bye.